Good morning, everyone. Thank you so much for the introduction. My name is Nerea Zabaleta, and I am an instructor in ophthalmology, and my lab is in Masaya Near. Today, I'm going to be talking about a vaccine platform that me, together with Dr. Luc Vandenberg, have developed. It is an AAV-based single-dose thermostable vaccine platform that provides durable immunogenicity. So the COVID-19 pandemic highlighted the need for improved vaccine solutions. Right now, mRNA vaccines are the gold standard of vaccines because they have great features such as development speed and high efficacy at peak of immunogenicity. However, as the pandemic evolved, some limitations became apparent. The most evident one is the waning immunity. As you see here in the graph, after vaccine administration, antibody levels decrease in the following months, and booster shots are needed to increase those antibody levels and protect against infection. Additionally, as the vaccines as they are right now have a suboptimal antigenic breadth because they are based in the original Wuhan strain. And as all of you already know, every few months during the pandemic, we have seen a new variant of concern emerge. And these variants escaped partially the immunogenicity provided by that initial strain. Additionally, mRNAs are not stable and they have strict cold chain requirements. And finally, as you probably have experienced, they are pretty reactogenic. So our solution is to use a different platform. In this case, it is going to be an adeno-associated viral vector or AAV-based genetic vaccine. AAVs have been extensively validated clinically and commercially, but in this case, we are going to use them in a vaccine context, and there are going to be several differences. So we are using a pro-inflammatory capsid. In this case, it's going to be an AAV serotype 11, and it doesn't have a relevant pre-existing immunity in the population, which will allow for the administration in worldwide. Additionally, the capsid is going to be carrying as a transgene a non-self antigen, which is going to be by itself immunogenic. The dosing is going to be pretty different to that in gene therapy. The doses are going to be much lower, 3 to 4 orders of magnitude lower, and they are going to be injected intramuscularly with a very focal biodistribution. And finally, the expression of the antigen is going to be self-limiting. As you see here in the figure, in mice, few months after the administration in the muscle, we don't see any transgene expression. So as a proof of concept, we have generated AAV-based vaccines for COVID-19. So it is an AAV11 that is carrying as a transgene the spike gene of SARS-CoV-2 virus. So this platform could potentially be adapted for other applications by just changing this transgene to expressing a different antigen. The closest example would be to use antigens from other respiratory viruses, such as influenza. However, we have the potential of having also potent vaccines, both preventative or therapeutic vaccines against other viruses that are not respiratory. For example, HIV, hepatitis viruses, or papilloma virus. And finally, we could also explore the generation of therapeutic cancer vaccines. However, as I said, we have done our proof of concept studies with the COVID-19 vaccine. So when we inject a single intramuscular dose of our AAV COVID vaccines in non-human primates, we see that in all of them here in orange, there is the development of a high level of neutralizing antibodies against the transgene few weeks after the administration. In fact, if we challenge these animals with the live SARS-CoV-2 virus, so if we inoculate live SARS-CoV-2 virus to these animals, we see that there is no replication of the virus in the upper respiratory tract or in the lungs, while in control animals, there is an ongoing infection, as expected. To note here is that we have near sterilizing immunity in the upper respiratory tract, and this is very different. This is much more protected than what we see with mRNA vaccines in the same model. So, as I said before, waning immunity has been a big problem in this pandemic. So we had to test the durability of our vaccines and see if AAV vaccines were able to induce a more durable response. So this is again in non-human primates, where we vaccinated 4 animals with 2 early candidates, and we looked at antibody responses over time. And we saw that up to 20 months after administration, so almost 2 years, animals maintained the peak levels of antibody responses, which is a great advantage of this platform. So AAVs are also genetic vaccines, same as mRNA vaccines. So we retain the advantage of being able to reprogram very, very rapidly the vaccine and with great results. So in this case, we wanted to produce more potent vaccines because we want to have as low a dose as possible to make sure that we are able to produce many doses faster. So we engineered our first generation platform. You have seen all the data with the first generation platform to express higher levels of the antigen by changing the promoter. So as you see here, the second generation platform that has a minimal CMV promoter that triggers a much higher expression of the transgene is able to induce higher antibody levels at the same dose compared to the first generation platform. Additionally, we have also engineered the transgene to generate different flavors of our vaccine that are going to express different variant of concern spikes. So as you see here, we have generated the Wuhan, Beta, Delta, and Omicron variant vaccines, and we see that all of them are able to induce equally high neutralizing antibody titers against the self-transgene. So this gives us a reason to believe that we should be able to swap the antigen as we like and have equally potent responses. So with this new second generation platform expressing beta, we did another challenge, another protection study in non-human primates. In this case, since we wanted to put our second generation platform to a more stringent test, we injected 10 times lower doses, and this is 10 to the 11 genome copies total. And we challenged the animals on week 7, which is 2 weeks earlier than before. So we see here that the animals are significantly protected in the upper respiratory tract and in the lungs. However, we do not see that near sterilizing protection in the upper respiratory tract as opposed to the previous experiment, but we believe this is because it was too early and animals did not fully develop the immunity. However, if we look at similar experiments with mRNA vaccines, we will see a very similar profile where the upper respiratory tracts are not as well protected as the lungs. So finally, we also wanted to see if we were able to manufacture our products at scale. So we partnered with Novartis, and they adapted their commercial process to producing 2 of our early candidates. And they were able to successfully produce them at levels similar to what they would expect with their commercial product. And we were able to have a recovery of final drug substance expected for AAV drugs. We also knew that AAV was pretty stable, so we did a stability study where we kept aliquots of vaccines at different temperatures, and we saw that aliquots that were kept at 4 degrees or 25 room temperature for 1 month were able to retain potency. So AAV is a vaccine platform suitable for single-dose administration, which is going to induce a potent and protective immunity, which is going to, and antibody responses that are going to last for long term, We also know that we are able to very fast and robustly adapt our platform to emerging pathogens. And finally, we have seen that we are able to produce the vector at scale, and it has minimal cold chain requirements. So the next steps are going to be to continue IND-enabling preclinical studies, to establish GMP manufacturing partnership to produce clinical doses. Right now, our estimates are that in a commercial at-scale manufacturing process, we would be able to produce 1E11 genome copy doses under $10, which is in line with a vaccine application. We would also like to expand our existing patent portfolio, which covers the use of AAV, specifically AAV11 serotype, in combination with our most potent promoter for several applications. And finally, we would like also to demonstrate proof of concept for other applications. We are currently actively working on a pan-sarbecovirus vaccine, which will protect against all the beta coronaviruses, including SARS-CoV-2. Thank you so much.